Hello, everyone. Welcome to our webinar on protein misfolding, folding and misfolding in context, brought to you by the Protein Society. My name's Liz Meyering. I'm the president of the Society, and I'm delighted to introduce our webinar today on this really important topic in protein science. Before turning this over to the leader for today's seminar, Caitlin Davis, I'm going to make just some brief announcements. First, if you have suggestions for webinar topics or would like to organize a webinar, please contact us. You can get our contact information at the website for the Society that's shown underneath the webinar logo. Our next webinar is going to be in February 12th, and our topic for that one is bacterial cell wall as a living biopolymer. So we welcome you to that as well. And I'd like to invite you to join us for our annual symposium next summer, which will be in San Francisco, June 26th to 29th. We have a fabulous program and lots of travel awards for young scientists. So you can find more information about that as well at our website, the Protein Society website. I also want to highlight the Protein Society journal, Protein Science, which publishes a wide range of exciting protein science, and I encourage all of you to publish your work there as well. And now to our main event. I'd like to thank Caitlin Davis for bringing together this set of researchers on this topic that has very far-reaching and transformational, I would say, fundamental and translational importance. A bit of background on Caitlin. She obtained her BSc in chemistry and mathematics from the University of Michigan, then did her PhD with Brian Dyer at Emory University on fast-folding beta proteins. She then transitioned to developing new methods to investigate proteins in cells. This is part of the in context that we're hearing about today. And when she was a CPLC postdoctoral fellow with Martin Grubela at University of Illinois at Urbana-Champaign. And then in 2020, she took up her faculty position in the Department of Chemistry at Yale University. She has been recognized with prestigious awards for her innovative research, and that includes an NSF Career and Beckman Young Investigator Award. So I'm very much looking forward to hearing her presentation today, as well as the other presentations that are very nicely complementary, as you'll see, to give a better understanding of this frontier topic of folding and misfolding in context. I'd also like to thank Stephen and Daniel for sharing their exciting research with us today. I'm really excited. It's one of my favorite topics. So before I turn things over to Caitlin, just a couple of logistical notes. The way we're going to have this webinar is that each speaker will speak for about 25 minutes or so. Then there'll be about 5 minutes for some questions. Following the talks, we'll have further questions in a discussion format among our speakers. Please take a moment now to find your Q&A tab. And in the Q&A tab, you can type your questions at any time. Only the organizers and speakers will see them, so the audience doesn't see your questions. Following the presentations, Caitlin will read out the questions. And of course, we can't answer all of them in real time, but speakers may also type answers to the questions in the chat and circle round to them again in a bigger sense at the discussion part of the webinar. And I'd like to thank everyone in the audience for joining us today. Your questions are really key for enriching the webinar, and so please do share your thoughts. And with that, I will turn it over to you, Caitlin.
Great, thank you. That was a really nice introduction to the topic and as well to me. So today we're going to be discussing protein folding and misfolding in context. And I think a takeaway that I hope that everyone takes from this is that there are still unanswered questions in the field of protein folding, and in particular in our ability to understand how proteins fold in cells. And so I selected speakers that represent both experimental and theoretical approaches to understanding this. And I'll present first on some individual examples of how proteins fold, and then Daniel will speak second about how he uses theoretical models to aggregate this information. And then Stephen also is an experimentalist, but he thinks more at the proteome scale, and we'll be able to put some of this in context. So with that, I will go ahead and share my screen. So today my presentation will be about mimetics of in-cell solvation for protein folding. So as I just stated, traditionally proteins are studied in vitro in dilute aqueous solution, but we know that only 60 to 70% of a cell's volume is made up of water. Another 8% is made up of electrolytes and small organic molecules and metabolites. And the other 22% is made up of large macromolecules. This is our nucleic acids and proteins. So inside cells, there's a tug of war between steric crowding interactions that are stabilizing and non-steric sticking interactions that are typically destabilizing. So in order to understand protein interactions, we really need to be able to carry out our studies of them inside cells. And my approach is to study protein folding in vitro and in cell hand in hand. So first, I characterize a protein behavior of interest in vitro in dilute aqueous solutions using an intrinsic probe of the protein behavior of interest. So here I'm showing you the intrinsic probe tryptophan. Then once we fully characterize that protein behavior, we create a labeling strategy that minimally perturbs the protein behavior of interest in vitro. We do this using large fluorescent protein labels or small covalent dye labels. Once we've designed this labeling strategy, we then introduce our fluorescently labeled system into cells. So this labeling strategy allows us to observe the protein behavior of interest in the native context in cells. This cycle is then closed when, after making the same observations in cells, we go back in vitro and modulate the environment to mimic and understand what we observed in cells. And so perhaps not all of this is accessible to everyone, so everybody isn't going to want to study every single protein in cell. It would be easier if we had ways to mimic what's going on in cells, and what I want to focus on today is this last arrow here. So what we've learned about how to connect what we observe in cells to in vitro experiments, so how we can better mimic or make better predictions about what we should be observing in cells. So in order to make these measurements in cells, I use fluorescence imaging. So this is an inverted epifluorescent microscope, and we can put our fret labeled sample on the microscope. And we can do this experiment in vitro. We can express our fret labeled proteins in cells or inject them in cells. And we've also done these experiments in whole organisms, in living zebrafish, and now we've done some work in tardigrades as well. We can perturb our equilibrium of folded and unfolded protein using temperature. So similar to a circular diachrosm or fluorescence melt, we can resistively heat our sample on the microscope and monitor as our equilibrium of folded and unfolded proteins change. So here I'm showing you some of that data as we raise the temperature from 20 to 45 degrees and compare fret in vitro and in cells. So initially we're starting in a folded conformation. So we have a lower donor over acceptor ratio. More acceptor tells us we're in a more compacted or folded conformation. As we raise the temperature, our protein unfolds. And then at higher temperatures, we sometimes observe aggregation or turnover in this D over A signal. Now, when we compare in vitro to in cell here, what we observe in general for proteins is that they tend to be more compacted in cells compared to in vitro. And we can explain this due to crowding. And that we also observe that they tend to be slightly stabilized in cells compared to in vitro, although this is not always true. But we're not just interested in the thermodynamics that we can get from this experiment, we're also interested in kinetics, and we can get kinetics associated with folding by coupling this to a laser-induced temperature jump. So here we couple a 2-micron fiber laser that rapidly heats the water in our cell on a timescale faster than the dynamics of interest. So we can raise the temperature from something like 30 degrees up to the midpoint and monitor as the protein populations relax to this new equilibrium at the higher temperature. The advantage of doing this experiment on a microscope is that we get both spatial and temporal information. So if you think about a protein that's been expressed in the cytoplasm of a cell, we can analyze this image pixel by pixel, or we could segment it into specific areas of interest, for example, the nucleus and the cytoplasm. So here I'm showing you the protein expressed in the cytoplasm. We're going to start in a more compacted or folded conformation. So we're going to be more red. And then after the temperature jump, our protein will be in a more unfolded conformation. And so that will change to more green. And I'll show you what this experiment looks like. So here we're inducing our temperature jump and we're monitoring as these protein populations relax to the new equilibrium at the higher temperature. What you're observing right now are the kinetics associated with folding and unfolding of the protein. And once things have flattened out, we're observing the equilibrium. We can then release that temperature jump and monitor as the protein populations relax back down to the initial temperature. And for a two-state model, we have a unimolecular system, and so we can fit this to single exponential kinetics. So once this is finished, it will go ahead and fit those here. And we also observe that we return to the same initial value here, which tells us that this folding process is reversible for the system. Okay, so I showed you some kinetics there, and for the protein that I showed you the thermodynamic data, we see that there's little difference in the folding dynamics between in vitro and in cell. So this suggests for this particular protein that classic in vitro measurements and buffer can adequately reproduce the folding dynamics and mechanism. But from our thermodynamic measurements, where we saw that we had differences in the compaction and stability, this suggests that structure-dependent measurements like catalytic activity should be carried out in the presence of Crowders. And I didn't tell you about the specific protein that I was talking about, but the protein was phosphoglycerate kinase, which is a glycolytic protein. And if you study the enzymatic activity of PGK in the presence of Crowders, it enhances its activity by about 50-fold. So this suggests that its activity in cells may be enhanced by macromolecular crowding in the native environment. So the takeaway from this is that the best in vitro approach depends on the question that's being asked. If you were asking a question about the folding dynamics of PGK, our traditional aqueous solution did a good job. If you're asking a question about catalytic activity or something that's more structure dependent, perhaps our traditional in vitro measurement was not doing a good job. So once we've collected this kind of data, we want to be able to mimic these effects that we observe in cell in vitro so that we can explain where they're coming from. And the two major effects that we like to think about are steric crowding interactions and non-steric sticking or chemical interactions. So steric interactions or entropic interactions arise from the rigid volume that a protein takes up in a crowded environment. This favors reactions that increase the translational and rotational freedom of a molecule, and it does this not by stabilizing the folded state, but by destabilizing extended unfolded states. So you can think about if you were in a really crowded subway system, you're not going to walk around with your arms out. You're going to keep them close to your body. A protein is the same way. It's not able to access those really extended states in a highly crowded environment. We can mimic this kind of crowded effect in vitro using inert macromolecules like PEG or FICL. The chemical interactions account for all interactions between proteins in the environment aside from crowding. So these are going to be our enthalpic interactions. And here I'm showing you the difference between the folded and the unfolded state of a protein in terms of the amino acids at the surface as well as the charge. And what you can see is that the surface of these proteins changes between these two states. And so these interactions are going to be highly dependent on the surface of the protein and their interactions between the environment in the folded and unfolded state. So we can think about different environmental interactions in these different conditions. So we can mimic the ionic strength of the cell by raising the ionic strength to better match the cell. So often we conduct experiments in buffers that are designed for the experiment we're doing and not with a lot of thought to the ionic strength of the cell. So this is a simple first step to try and replicate our cellular environment. Then we can do an experiment in something like dilute cell lysate. So now we're introducing these large macromolecules that our proteins may be interacting with in cells. And finally, we like to consider the lysis buffer that we've used to generate that cell lysate. A lot of engineering has gone into the generating lysis buffers, especially for immunoprecipitation that protect protein-protein interactions. And these incorporate some of those small organic molecules and short fatty acid chains that are natively present in cells and can be a kind of intermediate between these different environments. So we're going to take a look at how these different these different molecules affect the stability of PGK in vitro, and we wanted to contrast the stability of PGK, which is stabilized, to a protein that might be destabilized in cells. And so this is going to be the outer membrane protein, variable major protein sequence, like sequence expressed, or VLSE. Outer membrane proteins have to be unfolded and transported to the outer membrane where they fold and function. And so our hypothesis was that it would be destabilized in the cytoplasm of cells. And indeed, when we do the same experiments in cells that I described earlier, we see that VLSE is destabilized in cell compared to in vitro. And it also has larger D over A values. So it's in more extended conformations in cells compared to in vitro. And we wanted to see if our buffers could replicate these differential effects that we observe in cell. So the first thing that we did is we titrated in increasing amounts of our inert macromolecule. Here, this is going to be FICL. Closed icons are VLSE and open icons are PGK. And we're going to contrast that with a measurement made in 200 millimolar potassium chloride because the ionic strength of the cell is around 200 millimolar and it's predominantly made up of potassium. Measurements made in that lysis buffer, and then our measurement made in those U2OS cells where VLSE was destabilized and PGK was stabilized. What we see when we increase the concentration of Crowder is that both PGK and VLSE are stabilized. And this is consistent with crowding theory. This almost perfectly extrapolates to what we observe for PGK, but it can't explain the destabilization of VLSE in cells. We then titrated in increasing amounts of our chemical interaction mimic, the cell lysate, and what we see is that we get a differential effect here. So PGK is stabilized and VLSE is destabilized. And so this tells us that these chemical interactions are actually quite important for modulating the stability of our protein. And again, this can almost extrapolate out to what we observe in cells. These differences can't be explained by ionic strength alone, as this had little effect on the stability of either protein. What we did observe is that the differential effect on the stability of our proteins could be mimicked in the lysis buffer alone. And in fact, if we add the measurement at 150 mg per ml FICL, which is around the rate of macromolecular crowding that we expect in a eukaryotic cell, to our measurements made in lysis buffer, we almost perfectly replicate what we observed in cell. So I then took this a step further with a rotation student who did a series of experiments to test whether there was a mixture that could quantitatively predict the stability of both proteins in vitro. So they did a screen of different concentrations of lysis buffer and FICL, and were able to identify a mixture of 150 mg per ml FICL and 60% of this pierced lysis buffer that generated the in-cell stability of both PGK and VLSE. So this suggests that this is capturing both the steric crowding and chemical interactions that we observe in our U2S cells. And as I was starting my lab, I had some questions about this mimetic. First being, if this is just accounting for nonspecific interactions, could it be useful for other macromolecules like nucleic acids? Does it work for peptides and small proteins, which are predicted not to be as affected by crowding interactions where crowders are much larger than them? And third, can we understand when this kind of mimetic fails? So I know that this is a protein audience, but I think it's important to think about what's going on with other macromolecules in cells. So I will touch on this quickly. This is the first study that we did when I started my lab in 2020. And we used a model RNA thermometer that had been previously characterized in cells. And it had been shown to be stabilized by about 8 degrees compared to in vitro. So this thermometer is labeled at the 5' end by ATO 488 and at the 3' end by ATO 594. And we were able to show that indeed we can do these experiments in my lab to look at the stability of the RNA thermometer in the nucleus and the cytoplasm. And something that maybe you didn't appreciate when I was giving you these stabilities is that there's a lot of heterogeneity when we're making measurements in cells. So this is the kind of heterogeneity in the population and the stabilities that we're pulling out from measurements made in different cells in the nucleus and the cytoplasm. And this is because, unlike in vitro, every cell is a little bit different. They're in different stages of the cell cycle. They're undergoing different stressors. The environment is different. And this is going to give rise to changes in the stability of proteins between different cells. But what gave us confidence in our measurement is that in general, across all of the cells that we studied, the RNA thermometer is slightly less stable in the cytoplasm than in the nucleus. And that's what I'm showing across the bottom here. So then, using the same kind of mimetic strategy, could we produce the cytosolic stability that we observed for this RNA thermometer? We went ahead and we did the same kind of screen, here swapping out the lysis buffer for mammalian protein extraction reagent. Because this lysis buffer is optically transparent, it allowed us to do measurements on the unlabeled RNA thermometer in addition to the FRET labeled system. And what we found was, again, indeed, we found a mixture that was able to replicate our in-cell measurement, and this was at 20% MPER. This is a more aggressive recipe, or it includes more detergent than the pure lysis buffer that we had used previously. And 150 mg per mil of PEG here, and this was to be consistent with the previous study. We also validated that our original mixture of 150 mg per mil FICL and 60% pure lysis buffer was able to replicate our in-cell stability, so that was still consistent. So yes, in some cases, this kind of a mimetic strategy can reproduce the in-cell stability of a nucleic acid. And we also generated this new recipe that can be easily generated that's optically transparent. Then we wanted to answer the question, does our mimetic work for peptides and small proteins? So we took a look at this protein lambda repressor, which has been very well characterized in vitro. And our first strategy to study this 80 amino acid protein was to label it at the NNC termini by fluorescent proteins. And surprisingly, what we observed is that even with these large fluorescent protein labels, the stability of our lambda repressor was unchanged by labeling. So this is showing very good agreement here. We then made measurements on the stability of our lambda repressor in cells, and what we observed is that the donor over acceptor ratios were lower in cells, which suggested a more compacted configuration, and then it was stabilized by about 5 degrees in cells compared to in vitro. Our hypothesis here was that the stabilization is arising from chemical interactions and not crowding interactions, because we predicted that this protein is so small that it wouldn't be impacted by crowding from large macromolecules in the cellular environment. So then we again went back in vitro to try and understand what was going on with our system. And we do here we're going to take a look at these D over A values, or that end-to-end distance of our protein first, to understand what's going on. So as we titrate in increasing amounts of FICL, what we see is that our protein is getting more compacted. So this is telling us that this particular system is not, or that it is being affected by crowding. Our lysis buffer had the opposite effect on this on the end-to-end distance of this protein. So it becomes more expanded as we increase the concentration of our lysis buffer. When we look at the stability of the protein, though, we observe something different. So even though we're seeing that the protein appears to be more compacted as we increase the concentration of our inert crowder, we see that there's no change in the stability of the protein, which is not consistent with crowding theory. Instead, what we believe is happening is because we do have those large fluorescent proteins attached to the lambda repressor with a short linker, that we're able to push those fluorescent proteins together without affecting the stability of the lambda repressor within. In EMPER, what we observe is that the protein is stabilized as we increase the concentration of this lysis buffer. And again, what we see here is that we're able to replicate that in-cell stability at a concentration of around 20% EMPER and around 15 or 150 mg per mil, our macromolecular crowder. In this case, the lysis buffer alone would be enough to predict our in-cell stability. So this suggested to us that chemical interactions alone can predict the in-cell stabilities of our small proteins and peptides. But we wanted to see if this was true for other small proteins. And in doing so, we came across an example where this mimetic failed. So what can I tell you about when this won't work? And the example I'll tell you about here is Barnase. So the version of Barnase that we worked with is catalytically inactive, and it also has a mutation to it to decrease its stability to near physiological. And what we see is that when we use the same kind of labeling strategy, there's very little impact on the stability of this Barnase. We then looked at the stability of Barnase in the nucleus and the cytoplasm of the cell. And what we see when we do that is that in the cytoplasm, we don't see an increase in these D over A values, we see a decrease. This is consistent with aggregation upon unfolding at higher temperatures. And this appears to occur, or this onset of aggregation temperature is higher than the melting temperature in vitro. When we look at protein expressed in the nucleus of the cell, what we see is a destabilization of the protein. So we see that the protein unfolds at a lower temperature, and then we also see onset of aggregation at a similar temperature that we observed in the cytoplasm of the cell. Because there's a differential effect in the nucleus and the cytoplasm here, our hypothesis was that we weren't going to be able to reproduce these opposite trends using our simple mimetic. And indeed, when we did these experiments in different conditions, just using an inert macromolecule or salt or lysis buffer isn't able to generate these differences. We did some experiments in salmon sperm DNA to try and account for the increased amount of nucleic acids present in the nucleus as well. The only way that we were able to generate that differential trend was by conducting measurements in cell lysates. So when we perform a measurement with Barnase in a cell lysate generated from the cytoplasm or the nucleus of our U2S cell and dialyzed into our buffer, what we see is that we're able to reproduce those trends, differential trends that we observed in the cytoplasm and nucleus of the cell. So this suggests to us that weak specific interactions with proteins in each compartment modulate Barnase's stability, and it's not a non-specific effect that we could account for with our simple mimetic. Barnase is not a eukaryotic protein, it's a prokaryotic protein, and we wanted to see whether or not it was still prone to aggregation if we investigated its stability in a prokaryote. So we repeated this experiment in E. coli, and what we saw is that in E. coli, our Barnase is destabilized by about 15 degrees compared to in vitro, but that we don't observe that aggregation at higher temperatures. And because it was so destabilized, we actually went back to the wild type Barnase, catalytically inactive variant of that, and perform the experiment in E. coli on that system. And we were able to see there that variant of Barnase was destabilized by about 10 degrees in E. coli. And this speaks to hypothesis in the field of, I guess, in-cell folding, that proteins have evolved these weak interactions that stabilize them in their native environment. So if you take a prokaryotic protein and put it in a eukaryotic environment, that it will stick and not be as stable, or it will not freely rotate. And if you take a protein from and do the vice versa experiment and take a eukaryotic protein and put it in a prokaryote, that it would also not be as stable in that environment, and it would stick. And so what we see here is that perhaps this is speaking to Barnase having had some negative interactions in the eukaryotic environment that are not present in this more prokaryotic system. So what we think is that the mimetic fails when there are these weak specific interactions that modulate stability. And so I told you about a whole bunch of different systems that we've thought about in my lab, and that we have a mimetic that is commercially available, that can be a quantitative predictor of in-cell stability trends, but that still cell lysates are necessary when specific interactions in the cell are modulating behaviors. And with that, I'd like to thank the people that contributed to this work. So here I'm highlighting that Haejin Yu did the work on the RNA thermometer, Eddie Nab did the work on Lambda repressor, and Ume Tahir is doing the work on Barnase. And this work was funded by a grant from the NIH and from the public. And with that, I'd be happy to answer any questions.
Great talk, Caitlin. There are several questions on the Q&A. Maybe I can pick a couple and ask you. So one question, actually two, had to do with those fluorescent proteins that you attach to as probes, and the question of how they may change the interpretation of your data, and whether smaller is better when it comes to those probes.
Yeah, I think that's a great question, and that speaks to the design element. We are actually, for Lambda repressor, going back and repeating the experiments with small organic dye labels to see how they affect the stability of the protein. I don't think necessarily that smaller is better. So for PGK, for example, I know that the two fluorescent protein labels are actually better replicate native conditions than a single one, because a single label only has the protein that it's attached to interact with, whereas you get rid of some of those negative protein-protein interactions between the fluorescent protein label and the protein of interest by having two of them there. Our thought is that in a cell, every protein is surrounded by about 10 other proteins, and we're just making two of those are fluorescent proteins. And so that's what the protein would normally see in its native environment. When you put a small dye label onto the protein, the major effect that we've observed for PGK is that it does speed up the folding, so that having those large fluorescent proteins creates a sort of drag on the protein that slows its kinetics, but it didn't have a large effect on the thermodynamics of the system. I have seen some weird effects from dye labels. So the dyes themselves can be localization signals that send your molecules to different compartments. So when we label our protein or nucleic acid with Alexa 594, it directs the molecules into the nucleus. And we've observed that the dye Alexa 488 can get through gap junctions and in between cells. So there are some other effects that need to be considered when thinking about a small dye that also is non-native and charged. Ideally, we'd like to get to a point where we're not using any labels at all, which is something that we're working on in my lab, and I think also something maybe Stephen can speak to as well in his approach for studying folding.
Right. I'm going to ask one question of my own, which is when you mix several components together, do you find that your effect is additive? So in those matrices that you build with those effects, so is it to a large extent just the added effect of those both, or there is some synergy going on there?
Yeah, it's a synergy. So the effects are non-additive. Yeah, I have a backup slide, but go to the go to the paper. So the effects are non-additive. It'd be really simple if they were additive. And I think anything you do to perturb the system or many of the things that we do, for example, I alluded to as we put different labels on the system, they have non-additive effects. And so it's not simple just to predict what's going on as you move between environments because things are non-additive. And perhaps you'll be able to explain that better.
I'm not sure, but it's definitely an interesting finding.
Okay, great, thank you.
Yeah.
So with that, I will introduce Daniel Harries. He's a professor of chemistry at the Hebrew University, and he's going to present on when push comes to shove, macromolecular crowding beyond hardcore repulsions.
Thanks, Caitlin, and thank you for inviting me to this session, which is really very interesting. I'm learning and hearing some new stuff myself. Okay, so before I forget, I will acknowledge the people that have been working with me, the group, and specifically in this talk, I will present data from Yale and Yale's work, and also a lot of data from a collaboration with Gary Pilak that has been very instructive for us. Okay, so sometimes we know that we see some beautiful phenomena in our study. It is very elegant, and we are tempted to describe it in simple terms. But then when we actually do the experiment, we often find that things are a bit more complicated. And there are all these other considerations that we didn't take into account. So today's my talk, I will really concentrate on these things that we have been learning that we have to take into account so as to think about these ideas of what happens to macromolecules when they are in complex surroundings. And we're thinking about processes that we already heard about today, protein folding, but also processes like protein-protein interactions and other processes that don't actually even have to be in a biological context, like aggregation of particles in the presence of other added constituents into a solution. So we know that the temperature, pressure, pH, and other chemical potentials can affect these equilibria. And we often neglect these other solution components, salts, sugars, polymers, and other organic molecules that are present in the cell and are shifting these equilibria left or right, depending on the interaction with the solute. But how are they actually driving these changes in equilibrium? Well, this is something that was already discussed many years ago. One of these ideas is crowding. And this idea that we already heard about, that when you have a lot of confining, other confining molecules around the molecule of interest, there will be some restriction. Now, in this metaphor of a picture, we see that a lot of people are not the only thing that is crowding, but actually that there is some kind of a halo around them that is helping them crowd or make the crowding softer than it would have been. And I'll come back to that point later on in the talk. So where do we see this kind of crowding? So we already know that that this happens in cells. This image that is often shown when discussing crowding from good cell shows a cell that is full of these macromolecules that can take up to maybe 30% of its volume. And then there are all these synthetic systems where we see something similar. So we can see this set of colloidal particles in a solution with or without a polymer. And we see that when the polyethylene glycol in this case is added to these colloidal spheres, we see something that looks like a phase transition into a crystalline order of the colloidal particles. So this is something that is very general. It happens in biological, but also in completely synthetic systems. We also know that there are some extreme cases, the water bears or tardigrades that can survive almost anything, even outer space. They survive these harsh conditions by using solutes, proteins and sugars such as trehalose that can replace water. And in those cases, the surrounding environment is so dense that the inside of this creature becomes glassy. So there are some extreme cases where crowding or changes in the environment are really very dramatic. And what is really interesting with this creature is that it can, after its desiccation, as we see in this short movie, it can come back to life once this glassy state is diluted back to its physiological concentration. Okay, so what do we know from thermodynamics about this kind of crowding effect? This is something that already was resolved by Gibbs at the very early days of thermodynamics. What Gibbs showed was that if we take some kind of system where there is compaction, for instance, from this unfolded, let me just change it to this laser pointer, from this unfolded to this folded state, by adding solutes that are excluded from the interface of these states, the unfolded and folded state, we can destabilize both states, the unfolded and the folded state. But what is interesting is that because the unfolded state presents much more interface with the solution, there is a lot more of this preferential exclusion of the solute, or conversely, a preferential hydration of this unfolded state compared to the folded state. Stated in terms of thermodynamic properties, we can think about this change in folding free energy with concentration, and what Gibbs showed through what is now known as the Gibbs adsorption isotherm is related to the difference in this preferential hydration, or this volume from which the solute is excluded. And so this property, this change in volume of exclusion, is a really important property in understanding by how much there is a stabilization of, in this case, the folding protein in the presence of the excluded solutes. This is, again, a metaphor. We see that the little fish are excluded from the big fish, the sharks. And if Gibbs had it say here he would say that the shock are therefore stabilized because of this preferential hydration. So the first attempt to give a molecular level explanation of this crowding is due to these two scientists, Asakura and Osawa. And in the 50s, they suggested a simple model that is so simple that it held up for many, many years as the main explanation for stabilization in the presence of crowders. And the idea is that if we have some kind of big, in their case it was a colloid, we can think about a protein in the presence of some solute, and these two are only interacting through excluded volume interactions. There is some volume from which there is this exclusion of the co-solute from, in our case, the protein. And then by compacting, either bringing two of these together or going from an unfolded to a folded state, there is a reduction in this volume from which the co-solute is excluded. That means that there is more volume for the co-solute to roam in, and that is essentially an entropic, favorable entropic driving force to forming this more compact state. And the interpretation is then that the change in free energy should be proportional to this change in the excluded volume and the osmotic stress that is driving, pushing these two molecules towards each other. And because the osmotic pressure is linear in the concentration, at least at low concentrations of solute, then this change in free energy will also be linear in the concentration of the solute. So how much of this really holds up in experiments? It turns out that quite a lot, which made this theory very successful over many years. Basically, we're thinking about an effective force between the compact particles. And in this little movie, we just saw the equivalent in air for this driving force. This Magdeburg hemispheres that are held together by the fact that they are evacuated from air, and that drives the two hemispheres towards each other, even though there is no attractive force between the two hemispheres, just by the action of this pressure in this case. In solution, this just translates into an osmotic pressure. So here is an experiment. Results from experiments with a small mini protein folding and unfolding in solution in the presence of many different solutes. You see some are very small, like glycerol, some are very large, like this dextran. And we see that the free energy is almost linear for all these many solutes, which is one of the predictions of the Asakura and Osawa theory. Also, this idea that the larger the solute, the stronger this driving force will be, is also held up. The stronger or the larger the solute, we see the effect of stabilization becomes larger. So these are two predictions that made this theory very successful. And one remaining one has to do with the entropy. So does entropy really explain the stabilization of proteins in the presence of crowders? So to answer that, I'll just remind ourselves of what unfolding free energies look like as a function of temperature. So we usually have heat denaturation at some point, In some cases, we can also see a cold denaturation. The free energy crosses this zero line at 2 points, so we have this curved free energy plot. And now we can ask how this folding free energy or unfolding free energy changes when we add solutes to the mix. And they can change in different ways. And from the way they change in the presence of solute, we can decode whether this is an entropic effect, or whether it's enthalpic, or whether it's some combination of the two. We can also look at experiments, and this is an experiment from the PLAC lab, showing that indeed there is this change in stability as solute is added, in this case polyethylene glycol, at varying concentrations. And then we see that these curves indeed change, and they also cross at this point, which is a telltale sign that tells us in which where we should be looking for the effect in enthalpy or in entropy. So what we find is that in this case, the effect of polyethylene glycol, if it is acting with this SH3 protein, is acting through enthalpy. And that is kind of surprising. It goes seemingly against the Asakura and Osawa model, which blamed entropy for the stabilization. We can look at many other examples by looking at these entropy and enthalpy contributions to the unfolding free energy. We can then pass the different cases into entropically dominated effects that should follow what we know from Asakura and Osawa, or enthalpically dominated ones, which are the more surprising mechanism that it cannot be explained by the Asakura and Osawa model. And again, if we look at these SH3 proteins folding and unfolding in the presence of different polyethylene glycols over a very large molecular weight range, we see that in all cases, the effect is enthalpic. So what is really driving the stabilization of the protein is an enthalpy. And again, an enthalpy tells us that there is some kind of interaction between the Crowder and the protein that is stabilizing the more compact state. What we find when we look at many different proteins and Crowders is that in general, it doesn't always hold true, but in general, smaller solutes often stabilize through an enthalpic contribution, while large solutes, typically polymers, often stabilize through added entropy to the more compact state. And again, we can see this for quite a few different proteins and several different Crowders. We can also present the same information in these entropy and enthalpy plots, helping us dissect these different contributions. But really the question is, where is this coming from? And maybe at this point, also, what is the relevance? So one thing I can say is that, and we've seen this also in the first talk, is that these kinds of effects are also seen in cells. And it's not just in these synthetic or reconstituted systems that we see this effect of crowding and this contribution of enthalpy or entropy that is making a difference for the different solutes. And then we can ask, why should we really care about all of this? Why should we care if something is acting through enthalpy or through entropy? And there are many reasons, but I'll perhaps mention just two. The first is the temperature dependence. And the question is, will heating of the system help the crowding stabilization, or will it go against it and interfere with the stabilization we expect from the crowder? The second point is if the interaction is enthalpic, meaning there is some kind of soft or chemical interaction between the solute and our protein, then maybe crowders can have some kind of specificity. So maybe they can have different effects on different proteins, even though they themselves have the same excluded volume, and at least from the perspective of Asakura and Osawa, should not really have a very different effect on the proteins. So to answer these questions, we thought about a model that will be able to answer these questions through the perspective of solution theory. And I won't go into all the details, but I'll try and point out the main components of this theory that is supposed to supplement the idea of Asakura and Osawa about depletion force, and not be limited to this idea of entropic depletion forces. So the components, the main components of the model that we are using has three phenomenological parameters. The first one is the excluded volume, just the size of the solute and the solvent and the solute. And this can be measured through densitometry, because basically we're talking about the partial molar volume of the solute. The second parameter is this chi parameter that is known from solution theory, and it tells us something about non-ideal mixing. It tells us how a solute and solvent interact with each other beyond just mixing entropy. And this we can get from osmometry, from vapor pressure osmometry, and we can even pass this chi parameter into an entropic and enthalpic contribution by looking at the temperature dependence of the osmotic stress in solution. The final parameter defines the interaction between the solute and the protein, this purple line, and this is epsilon. We call it epsilon, and this is the only parameter that we need an experiment with the protein to decode. These other two parameters we can get from binary solutions. So as a theoretician, I'm allowed one slide with an equation. So this is mine. We're using a variation on the Flory-Huggins free energy. Basically, we're looking at mixing of two components that have different sizes. That is the solute and the solvent. We also add this non-ideal interaction that has the chi parameter in front of it. And to this theory, that is an established theory of solutions, we add this epsilon, which describes the interaction of the solute with the protein. So by fitting our data of the free energy of folding or unfolding, and using the two parameters from the binary solution experiments, we can fit the data for the folding of different proteins in the presence of solutes. So in this slide, I'm showing just two mini proteins. One folds into this alpha helix and the other into this beta hairpin. And we're looking at these proteins folding in the presence of all these different solutes. And we see that the theory can describe quite nicely all these different curves by just by using this single parameter epsilon. Now, if all things were equal, this epsilon should be very similar for the two proteins in the case of an entropic effect, because the only thing that would be important would be the size of the solute. But what we actually find is that there is a very big difference in this epsilon, the interaction between the solute and the protein, in the case of these two different proteins. And specifically, we find that for this alpha helical protein, the interaction is attractive between the sugar and the protein, while for this beta hairpin, it is repulsive. So it is excluding even more than and beyond what the excluded volume would give us. So this is already indicating some kind of specificity, because the same solute, even though we're looking at different proteins, the same solute is giving us a different effect. So that means that the soft interaction is giving a different signature for the two proteins. Another way of looking at it is looking at differences between these two proteins in terms of the different contributions to the free energy. We can look at the soft interaction contribution, the non-ideal mixing, and the excluded volume. And what we see is that while the non-ideal mixing and the excluded volume contributions are very similar in the two proteins, what is different in their interaction or their response to the presence of the added sugar is the soft interactions that are acting in the opposite direction in the case of these two different proteins. So can we then go a step further and say why this same solute, why is it interacting differently with these two proteins? And we have by now quite a good idea, and what we're finding more and more is that we should be looking at hydrogen bonding. And it's not even the hydrogen bond between the solute and the protein, but rather the changes that the solute is imposing on hydrogen bonding between water molecules in the vicinity of the folding protein. So just briefly, I'll show some of the evidence that we have for this involvement of hydrogen bonds between water molecules in the presence of the protein. So what we see here is the probability of finding a specific angle between two water molecules. And we're looking here at this peak as the one that is contributing to hydrogen bonding. And we see that while the interaction between water molecules, the probability of finding these linear hydrogen bonds goes up in the presence of a sugar, or in this case, a polyol, sorbitol. And the interaction between water and the solute is actually making weaker hydrogen bonds. We can look at this snapshot from a simulation that is showing us how this is happening. What is really happening is that lousy hydrogen bonds that are formed between water and the solute are sacrificed in favor of better hydrogen bonds between water molecules in the vicinity of the solute. We can then add the protein into the simulation and ask how the whole system of these three components, how is it acting and interacting? And we indeed find that in this case, the sorbitol solute is excluded from the vicinity of the protein, seen through these through these G of R's. And we can see it then in this kind of cartoon. What is the combined effect of the whole system? What we're finding is that water that is neighboring the protein is being modified due to those lousy hydrogen bonds that are formed with the solute that is sitting further away from the protein. So basically, it's something like a monolayer of water that is surrounding the protein. And these water molecules have stronger hydrogen bonds than just regular water. And since water then likes itself better than even bulk water, that means that there are less hydrogen bonds that are available for the protein. And that means that the protein is looking for interaction, specifically hydrogen bonds, elsewhere, and that is within itself. So the compaction of the protein is driven by the water becoming even more hydrophilic than regular water, so that the protein shies away from it and condenses into this more compact state. So basically, that's what we know so far, but we know that there are many things we still don't know. One thing is what happens with more realistic crowders, and hopefully experiments, as we've seen in the first talk from Caitlin, will help us answer that kind of question. Those same experiments may also help us to understand what crowding looks like when we have several components that have very different kinds of soft and excluded volume interactions with the protein. And also, we're interested in how other processes are affected by crowding, and that is processes such as oligomerization, aggregation, and even phase transitions that can come about through the action of these crowding agents. So I think I'll end with that and be happy for any questions.
Thank you. Okay, so we had several questions in the chat, and I will try and synthesize some of them into one cohesive question. So there were a lot of questions about how you determine like what the best composition of co-solutes is for your calculations. So, so there's a question about like, what's the effect of urea, or, is PEG the best osmolite, or should you be using other ones? So what is your perspective on that in trying to create a model for these systems?
Right. At this point, I guess we would, okay, so one of the things that we would really like to be able to do is exactly that, is to find combinations that allow us to stabilize proteins to the extent that either they are in cells, or if we're talking about some kind of technological application, whether we can design a buffer or an environment that will be the most conducive for protecting these proteins or increasing their activity. So the first point is that I think we have to think about tailoring environments for needs. So if we want to recreate, the environment, a cellular environment, it's one thing. If we want something that stabilizes vaccines or something like that, then we should probably think about something different. But then when it comes to the details, it becomes complicated because I guess of this question I asked you before about what happens when you add several components. And that is something that we're trying to look at right now. I don't have an excellent answer.
Yeah, what do you need from experimentalists?
Exactly. So what we can say, at least, is that what was traditionally assumed that for instance, polyethylene glycol or phycol are idealized crowders is probably not so simple as first imagined. So we probably have some interesting things going on there as well, and we should, be thinking about them as more interesting than we thought before.
Okay, and there was one other question that I wanted to ask that was given in the chat. So the question was that considering the cellular environment is extremely crowded, how do intrinsically disordered proteins manage to remain to be disordered?
Okay, so one thing, I mean, first of all, it's an excellent question, and I'm not sure I know the full answer to that, but some direction. So one thing is something that you mentioned in your talk. So size matters, and when it's an intrinsically disordered protein, maybe it's already. So maybe we should be thinking about smaller units within that protein as the affected entity, and then we don't necessarily have. We shouldn't maybe expect the same effect on those as we think about large globular proteins. That's one thing. The other thing that is really important is, I think, chemical interactions are really vital, especially for proteins that are marginally stable. So if it's easy to shift the equilibrium one way or another, then even small changes in the environment will be important. And in fact, these calculations and experiments I was showing with the mini proteins, basically you could think about them as intrinsically disordered proteins because their stability is marginal. So for the experiments, that was very nice because we could shift the equilibrium constant very strongly left or right, just by adding, not so much of the solute, just because they react very strongly to the environment.
Great. Thank you. So I think we need to move on to our final speaker, and it's Stephen Fried, an assistant professor of chemistry from Johns Hopkins University. And he's going to tell us that protein folding can still surprise us even after AlphaFold. So take it away, Stephen.
All right, thank you so much, Caitlin, for the introduction and for the invitation to be a part of this webinar series. I just want to quickly check, is everyone able to see my screen okay?
Yeah.
All right, looking good. All right, wonderful. So I might have to slightly disappoint because I guess Caitlin gave a bit of like a prelude to say that I'll be talking a lot about proteomics today. And actually, I'm going to do something in this talk that's a little bit different than our usual, which is less on proteomics and more actually on two specific proteins that, in essence, we discovered, or we thought we discovered, interesting behaviors about them through proteome-wide assays. And then we really wanted to dig deeper and follow up on them. And it turned out in both cases, they really had some interesting surprises in store for us. Okay, so by way of introduction, I think probably most of the attendees in this virtual space are aware of the amazing strides that have been made in artificial intelligence to be able to predict three-dimensional structures of proteins based on their sequence with really, really high accuracy. And I think I speak for most of us that the sort of advance of these tools in the last 3 years have been really staggering. And I would go further to say that in addition to being really powerful tools, they really provide the most, I would say, compelling evidence to support the original hypothesis that was put forward by Christian Anfinsen in the 1960s and 70s, that the three-dimensional structure of a protein is indeed fully specified by the information content that is stored in its primary amino acid sequence. But of course, as we also know, I think many of us in this room, is that protein folding is actually a very perilous sort of process. There are some proteins, typically these like small, soluble, single domain ones, that if you take them apart, they're quite competent at spontaneously locating their native conformation again. And of course, the physical picture that's generally invoked to explain this phenomenon is the idea of a funnel-shaped energy landscape in which the native state represents some global thermodynamic minimum. But any of us who have ever, tried to express or purify a protein have encountered, difficulties. There are oftentimes proteins that if you try to force them to refold from a denatured state, they simply fail to do this. They either aggregate or they form something that's incorrect. And this propensity tends to be higher for proteins that are larger or have more domains or are not necessarily in the cytoplasm. And so there's a wonderful quote that is attributed to Eugene Wigner, who is a great sort of condensed matter physicist and statistical mechanician active in the mid-20th century, at the very, very onset of when computers were first becoming available to perform simulations and calculations on a scale that scientists hadn't really ever seen before. And he commented somewhat obliquely that it's nice to know that the computer understands the problem, but I would like to understand it too. And I think that us as protein folding biophysicists or protein scientists more broadly, I think that now we're sort of faced with a sort of very similar question, which is that it's nice to know that the computer can fold a protein, but the protein would like to be able to fold itself too, meaning that it's great that if you have this sequence, a computer can tell you what the right final answer is, but how come it's the case that when the actual physical molecule is trying to do the exact same thing, it makes mistakes, it gets degraded, it gets ubiquitinated, it gets turned over, it gets mislocalized to the wrong compartment. So it's kind of funny that we now are in this state where we know what the right answer is in many situations based on learning from the vast troves of data that are accessible on the PDB, but we don't always know, or at least we generally don't know, how it is that proteins are able to find that way and why they get lost in that sort of journey to their native state. Okay, so we're going to play a little bit of a game. This is called Two Lies and a Truth. So two statements on the slide are a lie, and one is a truth. And I didn't make it particularly hard. I think the one that's true probably stands out pretty obviously. But I think the other two statements that are on this slide seem like they probably ought to be true as well. And actually, they were things that I certainly thought up until maybe about a year ago. And I'm going to present to you some results from my lab that we've sort of stumbled upon recently that I think go against some conventional dogmas about protein folding, and I think are just examples of how there's a lot of interesting things to be learned when we sort of look at, even old systems, through a new set of eyes. Okay, so to give a very, very quick sort of summary of what type of research my lab has been doing over the last 4 or 5 years, we've been really interested in using methodologies in structural proteomics to look at protein folding from a proteome-wide perspective. And the experiment that we got pretty good at over years of iteration are these experiments in which we couple a global unfolding refolding reaction to a readout in the form of limited proteolysis mass spectrometry. So in essence, what this amounts to is we take cells, we lyse them carefully using cryomilling in order to get the proteins out in a native form, at least in as much as possible. We add guanidinium chloride to a concentration of 6 molar, wait overnight, let the whole proteome unfold, come in the next day, dilute 100x in order to initiate a refolding reaction. Some proteins, you might imagine, will succeed in this task of navigating back to their native state. Other proteins might not, and instead aggregate or form some soluble misfolded form. And then we can probe that outcome by performing pulse proteolysis with proteinase K, the idea being that if a protein can refold, it really should have no memory of the fact that it was ever unfolded, and therefore proteinase K will cut it in the exact same places. On the other hand, proteins that fail to find their way back to their native conformation, we might expect that misfolded ensemble to get cleaved by proteinase K in a different way that will result in different peptide products, which we can then in turn sequence and quantify using liquid chromatography mass spectrometry. Okay, so to summarize, I guess, like some of that work, or at least like one little portion of it, we have found in general the proteins that are involved in glycolysis, sort of one of those key metabolic pathways that we learn about in sophomore biochemistry, do not refold very well at all. So in E. coli, of the 9 glycolytic enzymes, only one can refold, and that's triose phosphate isomerase. The other 8 don't. One particular enzyme that we actually heard about from Caitlin, phosphoglycerate kinase, of the 110 sort of peptides that we were able to identify for that protein, 32 of them were altered upon this unfolding refolding treatment. So we would consider that a pretty high number for most of the proteins in our assay, even the ones that we don't consider the refolders. There would normally be a lot fewer than that. And the same is true in yeast. Again, only one of the 10 glycolytic enzymes can refold, in this case as hexokinase. And once again, PGK was a highly altered enzyme. And so we kind of decided that, should we really believe all these lip-MS stuff? This is a question that we get, right? It's obviously not a method that has been kind of a conventional, traditional one used in protein folding biophysics. And so the kind of initial motivation for this research is, well, let's take a protein that we're pretty sure does not refold from 6 molar guanidinium, namely PGK, and do the thing that biophysicists have done for a long time, which is purify and just check out and see what it does, and just really give ourselves a sanity check of should we be believing these like structural proteomic assays in the first place. Okay, so first of all, we can purify it. It's a very easy enzyme to work with. You can get it to high homogeneity and yields without a whole lot of work. We can do circular dichroism on it, and here you're sort of seeing that the native enzyme that's in black, or letter N, basically has a sort of conventional spectrum for a protein that has a high amount of beta content, as PGK does. If we perform a treatment that I'll call the U24R1 treatment, which means we unfold it for 24 hours in 6 molar guanidine, and then dilute it 100x and allow it to refold for 1 hour, you can see that actually the CD spectrum is sort of slightly different. I mean, it still seems to show a signature for beta content, but you can see that it does not overlay on top of the native one. Interestingly, and this will become more important in the following, if we actually perform the unfolding step for only 2 hours rather than for 1 hour, we actually find that that does refold relatively well, at least on the basis of circular dichroism. You can see that the purple trace overlays with the black trace pretty well. Now, another way, so you might sort of be looking at the CD spectrum in blue and saying, well, Stephen, that's a pretty small difference. Like, is that really a protein that you're going to say is non-refoldable just because there's a slight different, feature at 220 nanometers? Well, the answer to that question is definitely yes. If we do another sort of measurement of nativeness or refoldability, in this case, we're measuring enzymatic activity. So what we've done is we've taken the enzyme, unfolded it, refolded it, and then measured how much enzymatic activity it has left. You can see that indeed, this U24R1, this protein that has been unfolded for 24 hours and then allowed to refold for 1 hour, really is very distinct from the native, and that it's almost catalytically dead. And that protein that was only unfolded for 2 hours, consistent with the CD spectrum, is actually quite has native levels of activity. And I want to take a little moment to sort of interject that I think that CD, and actually probably a lot of other biophysical methods, probably actually underestimate or I would say sort of underestimate how well proteins don't refold, in the sense that there's a lot of proteins that probably can sort of, recompact, reform secondary structure, and therefore recreate sort of things that look like it's folded in terms of having, sort of a higher Fred or more dichroism. But if you actually do a very stringent test of whether or not it's native, and I think that enzymatic activity is probably a more stringent test, because really the protein has to be in a fully native structure in many cases to exhibit, high enzymatic activity. We find that that they don't necessarily sort of refold as well as we might think they do. Okay, now here's where actually stuff gets very interesting. So now what we're going to do is we're going to take this PGK, and we're going to now subject it to a conventional circular dichroism based measurement of the melting temperature. So basically, we take the protein, stick it in a cuvette, put it in an instrument, a CD, measure the dichroism, and step up the temperature by 1 degree every 5 minutes, and then re-record the spectrum. And what you can see is that the native state of this protein sort of shows a conventional sort of melting curve with a TM that's around 60 degrees. That's pretty common for a mesophilic protein, such as PGK from E. coli. But kind of strikingly, this misfolded protein, U24R1, the one that I told you has actually almost no enzymatic activity, it's also almost unmeltable. So it has a much higher sort of apparent thermostability than the native protein that it came from. And if you're wondering, maybe that's because you've just caused it to aggregate or form some amyloid or form some other like multimer, I can tell you that's not the case, because we also did analytical ultracentrifugation on these samples, and you can see that they basically, all the traces overlay on top of each other. So in essence, U24R1 is a soluble monomeric misfolded form of PGK, which has greater apparent thermostability than the native form, although has almost no enzymatic activity. So then the question is, well, what is this? Well, our best hypothesis right now is that this represents a population of an entangled state, or more specifically, what the U24R1 form of PGK is that it possesses a gain of a non-covalent lasso entanglement. So this is a topological form of misfolding in which sort of one part of a protein wraps through another part of the protein that's closed by a so-called non-covalently bound loop. And this is a form of misfolding that has now been extensively sort of studied and characterized computationally by Ed O'Brien and his very talented team, including his postdoc, Yang Zhang, who sort of led this, sort of computational work on PGK. And Yang found that in sort of computer simulations in which PGK was globally unfolded and then compelled to refold, that these types of, knots occur within the protein. And although we don't have direct experimental proof that this is the case, because we haven't been able yet to get a cryo-EM structure of the misfolded PGK, we are at least able to say that the ensemble of different misfolded states that Yang proposes in his computer simulations are in fact well explained by these types of structural proteomic experiments that our lab is good at, like limited proteolysis mass spec and cross-linking mass spec. So this is sort of just a heat plot to show that if you basically invoke 3 of these potential forms of entanglement, that some of them actually do quite a good job of explaining these like structural features that we see in the U24R1 form of PGK. So I think like the obvious question that this begs is, well, how is this happening, and why is it that the protein can refold relatively well if it's only unfolded for 2 hours, but now all of a sudden when it's unfolded for 24 hours, now it's like struggling in this kind of unusual way? And I think that the best sort of evidence that we have that kind of like speaks to that question is we did analytical ultracentrifugation, but this time actually looking at the unfolded forms of the protein. And what we found was something that was really surprising, which is that when you let the protein unfold for a longer period of time, according to AUC, it actually becomes more compact, which is definitely the opposite of what you'd expect. You'd normally expect that like maybe the protein just takes a long time to unfold, and it takes 24 hours to fully unfold it. But what we think is happening is that actually the protein is basically unfolded at 2 hours, but then if you go for a longer period of time, the more time it spends in this unfolded ensemble, it basically eventually sort of locates a state that creates these entanglements. And then when you release that unfolded ensemble from denaturant, those entanglements then get entrained within the native structure, therefore sort of locking them in and creating this kinetically stable misfolded form. So this is a model. I don't want to necessarily say that I think this is, the truth. I think that the reality is probably more complicated than what this model is showing. But this is kind of like, I think puts a picture on the words of what I've been saying, and at least is a model that's consistent with all the data that we have to date of what happens with PGK, which is that when you unfold it from its native state and you only let it stay denatured for a brief period of time, dilution from that unfolded state can result in something that is reasonably competent at refolding and going over this sort of like reasonably sized barrier, but sort of long periods of exposure to denaturation result in this unusual type of unfolded state that has an altered topology, and then release from denaturant from that state then puts you in a different sort of region of the energy landscape that tends to sort of trap you into these highly kinetically sort of trapped states that are not active, but which still contain many features that are native-like. And so I think there's an interesting implication there, right? Because if this is a form of misfolding that can happen in our cell, then I think we should be pretty worried about this, right? Like these are probably misfolded states that will be very hard for the cell to get rid of or recognize or degrade. And so I think that sort of being more aware of this type of misfolding, certainly in my view, kind of opens up a bunch of new questions. So to go back to our original game of two truths and a lie, I think we can conclude that it's not always the case that misfolded proteins are less stable than the native form. Okay, so in the second sort of like portion that I'll maybe go a little bit more quickly through, let's talk a little bit about chaperones. So chaperones are, of course, these molecular machines that cells have that sort of assist protein folding. And in E. coli, we have really three primary chaperone systems. So we work with, we have GrowEL, which is the famous, textbook one. It's this chaperonin known as HSP60. It primarily acts post-translationally, although it can also act co-translationally. And the key features of GrowEL is that it is both an unfoldase and a foldase. So that means that if it sees a misfolded protein, it can basically undo the mistake. So it can basically unfold a protein that has misfolded, in essence, kind of like reverting that mistake and giving the protein another chance to get it right. So so-called iterative annealing. But the chamber of the GrowEL chaperone is also able to facilitate folding. And so in that sense, it can act as a foldase. DNAK is the name that we give to HSP70s in bacteria like GrowEL. They also burn ATP, and they can also have unfoldase activity, but they don't create a sort of compartment within which folding can occur. And then probably one of the more curious chaperones that we have in bacteria is this thing called trigger factor, which we don't really have equivalents of in eukaryotes. It does not burn ATP. It is exclusively co-translational because it basically is almost a part of the ribosome. And it basically sort of monitors nascent chains that are coming out of the exit tunnel of the ribosome, and it's believed somehow, promotes the correct folding of the nascent chain as it's emerging from the exit tunnel. But the way that it does that has not been very, very clear over the last few decades. Okay, so we do a little bit of proteomics because we like proteomics. In this case, it's our sort of favorite LIP-MS experiment. But in this case, what we're not going to do is our typical kind of unfolding, refolding thing, but rather what we're going to do is just grow up 3 different strains of E. coli, one which is wild type, one which is deficient in DNAK and its co-chaperone DNAJ, and one that's deficient in trigger factor via just genetic knockout. And then what we're going to do is grow up these cells and then get the proteins extracted from them, and then probe those conformations with limited proteolysis. And basically, it just asks the question, which proteins endure a kind of a permanent structural alteration on account of the fact that their biogenesis was not able to enjoy the service of that particular chaperone. And what we find is that there are quite a few proteins in E. coli that depend on DNA K. So about 27% of the proteins that we have data for are really altered in the presence of DNA K. And actually that number is a really small percent for trigger factor. And I would say that very, very broadly, these are numbers that are consistent with phenotype. We know that if you knock out DNA K, E. coli is really not very happy with you. It is viable, but it does grow very slowly. Whereas if you knock out trigger factor, there's actually almost no phenotype. The cells basically grow the same at pretty much every temperature. So the fact that there was not so many, proteins that were getting altered in the delta trigger factor background did not necessarily surprise us that much. And this is just a contingency table to say that, like, of the proteins that were perturbed when we removed trigger factor, which wasn't very many, most of them were also perturbed by DNA K. So basically, there's kind of a small subset of proteins that are maybe very greedy and just like to have, lots of chaperones in order to help them fold because their folding is more complicated. But we really wanted to understand, okay, so who really does seem to care about trigger factor? Like, not that many proteins seem to care about it, but one protein that, at least according to our proteome-wide assay, did care about trigger factor was GAP-A, or glyceraldehyde 3-phosphate dehydrogenase. And the reason why we gravitated to this particular protein is because it is another glycolytic enzyme, and our lab just seems to really like glycolytic enzymes these days. Now, I'll point out, not that many sort of sites within the protein were altered in the delta TIG. It was only 3 out of 100, but we still thought, okay, like, let's just, see what's going on with this protein and what happens when we remove trigger factor. Okay, so here's the experiment. What we have are these 3 different strains of E. coli, right? Either wild type, delta TIG, or delta DNA KJ. We're going to use CRISPR in order to edit in an epitope tag to the C-terminus of GAP-A at the native genomic locus. And the reason why we're going to do this is because, since we're deleting chaperones, we don't think it would be a very good experiment to start overexpressing the protein on a plasmid, right? Because then you're sort of, it's like a double whammy on the cell if you're deleting a chaperone and forcing it to overexpress some random protein, right? So what we want to do is like a more sort of careful way to really interrogate the structural changes that a protein endures in its native context. And the way that we're going to do that is to basically just put an epitope tag directly on the chromosomal copy, in this case of GAP-A. And then for each of those 3 strains of E. coli, we're going to grow them up, either wild type or in the chaperone null backgrounds, lyse the cells, purify the proteins, and then do various forms of characterization on them. So what do we find? So the first experiment that we did is sort of classic in folding biophysics, circular dichroism. So what you're seeing is the CD spectrum of the wild type, all these are wild type, these are all wild type GAP-A. It's just whether or not the cell was wild type or delta DNA K or delta trigger factor. And what you're seeing is that actually, if I get rid of DNA K, the CD spectrum is basically superposed. That's consistent with the experiment. Like, there was no difference in the LIP-MS pattern for GAP-A between wild type and DNA K. But the result for trigger factor was really, really, really weird, in that what we were expecting was that maybe this protein would be, sort of have a smaller circular dichroism or a less pronounced sort of, feature. And in fact, it was the exact opposite of that. What we found is that in the absence of trigger factor, GAP-A actually shows a much more prominent circular dichroism feature than the wild type form. And not only that, if you get rid of trigger factor, the melting point of GAP-A goes up by about 5 or 6 degrees. So the protein actually has greater thermostability if it is created in the absence of a chaperone. And if you thought that was weird, when we measure the activity of the enzyme is also boosted by a factor of 4 or 5-fold when it's made in a cell that is deficient in a chaperone. So all of this was getting us very confused because, of course, chaperones are supposed to help protein folding, not obstruct protein folding. But that was sort of where the data were leading us. And so we really wanted to know, what is this relationship between GAP-A and trigger factor that seems to be going against at least our conventional thinking of what a chaperone is supposed to do? Oh, and sorry, one other thing, we also did analytical ultracentrifugation, and again, just showing that actually here, you can see that there is a slight difference in the sedimentation coefficient between the delta TIG and the wild type. So again, there does seem to be an actual structural difference between this protein, but it's not so big so as to make us think that the protein is like aggregating or forming different oligomer states. I will point out here that GAP A is actually a tetramer. So you can sort of see that there's a much smaller population of the monomer, dimer, and trimer, but most of it is in fact occupying the sedimentation coefficient that you would expect for the tetramer. So what is going on here? And I guess in order to answer that question, the first question that I guess we should all be wondering is, well, what does trigger factor even do? Because it's kind of like a weird chaperone, doesn't burn ATP, it's only in bacteria. Like, what does trigger factor even do? And what I would say is that we have benefited from a close sort of dialogue and collaboration with Bernd Buchau's lab in Heidelberg, who I would say in the last few years has come out with a set of experiments that's actually given us for the first time a clear mechanism of what this chaperone does. But before I can sort of teach you about that, I need to tell you a little bit about various types of co-translational assembly. So proteins, as it turns out, can assemble into their sort of native assemblies when they are still being synthesized on the ribosome. And that sort of co-translational assembly goes under a number of different varieties. We can either have so-called co-post assembly, which is assembly that involves a completed protein chain, then going back to the ribosome and interacting with a nascent copy. And then we have also co-co assembly, which is basically an interaction forming between two nascent chains. And this happens most prominently in Homo multimers, because of course the ribosome itself creates, or the polysome itself creates, an assembly line that can promote two nascent chains coming together that are being translated from the same mRNA transcript. Now, what experimental methods allow us to interrogate this type of co-translational assembly? So the main tool we have at our disposal is ribosome profiling, also known as RiboSeq. And there's sort of two particular flavors of ribosome profiling that can be useful for looking at assembly. One experiment, which is called selective ribosome profiling. In this case, we have cells, we break up the mRNA with an RNase, and then we isolate these individual ribosomes by purifying them against a particular sort of target. In this case, it's going to be trigger factor, but it could be anything. And then what you do is you map the reads that come from those ribosome protected footprints, and you look for what are sort of reads that correspond to regions that are highly enriched in the purification fraction as opposed to the non-purified fraction. And that basically tells you regions within the codon lengths where that interaction starts to occur. Another way of doing this experiment is known as disome-specific profiling. In this case, what we're doing is we basically take advantage of the fact that if two nascent chains are interacting with each other, then what they'll do is they will cause two ribosomes to stay stuck to each other even after you have cut the mRNA with an RNase. And then if you use a sucrose cushion to isolate that sort of peak that corresponds to the double weight of a ribosome, and you basically look at what reads are enriched in that disome fraction, you're basically able to see at what part of a protein do we start to have co-co assembly occurring. And these were methods that were pioneered by Buchau and co-workers. So what happens is, here's an example of a protein in E. coli, NRDB, which forms an obligate homodimer. And what you can see is that trigger factor starts to talk to this protein when we have about 120 to 200 codons translated. And the protein doesn't start to undergo co-co assembly until we have more like 300 codons translated for it. And the key experiment is what happens when we knock out trigger factor. What you can see is that yellow peak shifts dramatically to the left. And in particular, it shifts to a portion that was actually sort of occupied by trigger factor itself. So in essence, what this is telling us is that trigger factor basically is blocking the protein from undergoing co-co assembly, and that if you get rid of trigger factor, these nascent chains can now start assembling with each other much earlier on during co-translational folding. And this effect is not just true for NRDB. Here's a sort of metagene profile that shows that actually there's a bunch of other proteins where this is true, where the deletion of trigger factor basically shifts to the left the onset of co assembly and moves it closer toward the portion where trigger factor would have otherwise bound. So the model is that what's going on here is that trigger factor basically prevents premature co assembly and sort of promotes co assembly happening at the appropriate time. Now, what happens if we look at GAP-A? Well, here's an AlphaFold 3 model of GAP-A, which is a tetramer. And what I'm showing you in red are the last 30 residues of this protein. And what I want to call your attention to is that the dimer interface that allows these two protomers to come together actually involves the very, very, very end of the protein. And this actually creates a little bit of a problem, right? Because if we want this protein to assemble co-translationally, and it needs that very C-terminus of the protein in order to do that, we're not really giving it a whole lot of time on the ribosome to be able to get that job done. And this sort of amazing, experiment that, basically I think really gives us a much better clue of what's going on here is that we find in the case of GAP-A that trigger factor binds very, very tightly to it. And then when trigger factor is absent, we do in fact get co-assembly happening at the very sort of end of translation in the last sort of 30 codons. But if we look in wild type cells, we find that that co-assembly signature has completely been lost. And so in essence, what these data show us is that what trigger factor is doing is that it's doing its normal job of trying to prevent co-assembly from happening too early. But because for this particular protein, the assembly occurs so close to the end, the protein basically then gets forced into assembling in a co-post manner. And at least if you believe our activity and our CD assays and everything else, apparently this is a protein that does not want to fold co-post. It would much rather fold co, but trigger factor is sort of getting in the way. And so in essence, what our hypothesis here is that biology is faced with a catch-22. Trigger factor assists the assembly of some proteins, but at least for one case for which we have data, it obstructs the assembly of another. And I think that actually this is a pretty interesting sort of finding. It has some pretty, I think, broad implications. More narrowly, one sort of interesting implication is that we found that if you look at the abundance of different glycolytic enzymes across the tree of life, what we find is that glyceraldehyde 3-phosphate dehydrogenase is generally one of the more abundant glycolytic enzymes, but it is way more abundant in bacteria than it is in eukaryotes and archaea. And it's only bacteria that have trigger factor, okay? So our theory is that trigger factor had such a negative effect on the folding of GAP-A that it basically forced all bacteria to make like 4 times as much of this enzyme to make up for the fact that its specific activity is 4 times lower. And I think more broadly, the implication is we normally think of it as a good thing that chaperones are able to recognize a wide array of different clients and help out a lot of different proteins. But there's also sort of a liability that comes with that because chaperones tend to have a relatively broad specificity, that means that they don't always bind to the proteins that they are supposed to, in quotes, bind to. And I think that is an interesting sort of area that has a number of implications for disease and health that we want to explore further. So to go back to our little checklist, we could say it is not always the case that chaperones help proteins fold. We hope they do, and they probably generally do most of the time. At least there wouldn't really be a reason for them to be around evolutionarily, but there do seem to be these kind of examples of a cost-benefit trade-off, so to speak. Okay, so I probably spoke for too long, so I'll stop right there. I'll just once again acknowledge the individual. So everything I told you about PGK was from April Shah in my lab. Everything you heard about GAP-A is from Divya Yadav in my lab. You saw a lot of work on analytical ultracentrifugation. Wouldn't have been possible without Barbara Amon and Karen Fleming. Computational work from Yang Zhang and Ed O'Brien's lab. And of course, like we were staring at these data for a very long time, thinking that we were going crazy until we had an amazing call with Baron Bukau and coworkers who have really given us, I think, a much better idea of what's going on in that case of gap A. So with that, I'll end and hopefully take some questions.
Great. We have a question in the chat. So the question is, for the PGK enzymatic activity experiments, can you speak to whether keeping the protein in guanidinium chloride for 2 hours compared to 24 hours disrupts ribosomes and thus shifts the population to the entangled state due to less co-translational protein folding, or are you inhibiting translation after lysis?
Yeah. I'm sorry if I was unclear. So that it's like very simple, everything in vitro. So for that particular experiment, we've isolated PGK that was overexpressed on a plasmid in a wild type BL21 cell. All the unfolding and refolding was done in a test tube. And so, yeah, there's no sort of signature of co-translational folding. Although I do think it is like worth pointing out that we do hypothesize that proteins like PGK, which cannot refold well, and I didn't show the data for it here, but they don't refold well even if you give them chaperones. We think that proteins like that tend to have a higher propensity to obligately fold co-translationally. But the data that I showed here didn't directly show that. That's more of a hypothesis we have.
I have a question.
Yeah, I have a question after you. Go ahead.
So I think that, these talks have really nicely illustrated where we still have really important work to do. So thank you all for that. But my question is this. So what we've seen often with calorimetry actually is when we heat and cool and reheat, little proteins, they often go to something that has a higher TM, believe it or not. So I'm thinking that these phenomena that you're talking about are of alternate folding, right? Either in the system that you had, Stephen, but also it relates to the other talks as well. This can be way more common. We just haven't caught it.
Yeah, I think that like if you'll allow me to like stand on a soapbox like for a little bit, I think that we in biophysics have a little bit of like, a romance with like reversibility, right? Because it allows us to use familiar equations that are very powerful. And of course, it's not based on nothing. I mean, there are a lot of FRET experiments and CD experiments that you can do where if you do the right controls, it will basically look like things are reversible. And I think the bottom line is that there's a lot of ways that a protein can kind of, blob itself together that will put two FRET labels close to each other, or that will create alpha helices and beta sheets, but that doesn't necessarily mean that it's the same thing. And so I think that assays like activity and probably also like, thermal signatures are probably going to be better at actually really giving you a true sense of what's reversible. And of course, if something's not reversible, then you can't use your equations, and then, it makes it harder to write a paper. So that's kind of why I think this is a feature that hasn't always been sort of like, discussed. But I would agree with you. I think that it's probably more pervasive than we would generally think.
Thank you.
So I have a question that is not something that you covered at all, but came to mind. So I like glycolytic proteins, but I know that you tell me they're bad at folding. So that stinks. But is there anything that we can learn about the ones that are good at refolding? So like, are they part of metabolic pathways that are important to recover first in response to stress? And then there's time for the other proteins to be like expressed later and recovered in that way? Like,
I think I'm glad that you asked that, Caitlin. Like, I think that does kind of give me an opportunity to sort of say something that I think is true, but which I don't know is true. But it makes sense, which is that proteins that need to be good at refolding, right, from like a biological perspective, are ones that basically have to like, come in and out of like different compartments, right? Because oftentimes when you move a protein from like, let's say, the cytoplasm to the ER or to the mitochondrion, those pathways oftentimes involve unfolding the protein in order to like pass it through, like, let's say, a
I think he froze.
I'm like, am I frozen or Steven's
I
frozen?
think Stephen's frozen.
Oh no.
I don't think he knows that, of course.
No, neither do I. While Stephen is frozen, there are a few of you that are trying to answer or ask questions, either by raising your hand or maybe trying to type in the chat. If you could use the Q&A to post your question, that will allow Stephen to answer them after the session is ended. So there were a couple of you. And then if he makes it back, we can try and ask them as well.
I have another quick question, because this
Mhm.
goes to ones that were in the chat before too, about sort of the modeling aspect, right? So coming back to I'd like to know more what you think, you and Daniel, about the modeling aspect. Like, where will the modeling be taking us? You know, like, how can it help us better get at this? There's so many kinds of modeling, of course. There was a question about the force fields, and these are things we still need to work on. But I wonder if you guys have comments on the modeling aspect. Because we
Right.
need
So,
to explain to people what AlphaFold can and can't do. Do you know what I mean? Like, this is important.
so this thing with AlphaFold is so important because it's predictive, right? So the modeling, for at least for the crowding, it's always been a theory in hindsight. So being predictive is kind of the goal, and hopefully when we have enough information. So the point with AlphaFold is that you train this entity to give you the folding. I mean, it would be really great if we have something similar for crowding effects.
Mhm.
Sorry about an internet glitch. But yeah, I mean, I think that AlphaFold is good at what we train it on. Like the PDB is
Oh no.
Oh no.
Stephen's internet decided there was a hard cutoff to his talk. I
Caitlin,
think,
do you
yeah,
want-
so I think my perspective on AlphaFold is it's a great tool for making predictions, but we're not yet at the point where, if we're thinking about thermodynamics and kinetics, that AlphaFold can predict something like that, or what mutations do to a protein, like a single point mutation, how it alters its thermodynamic stability, and how that's going to be affected as you move between environments. So there's just a lot that's not known. Sorry, Stephen, you're going to try somewhere else?
yeah, I'm gonna try to like decamp from my office and see if I can get a more stable connection. I'm sorry about that.
This feels like a behind the scenes at SNL tour.
Exactly.
Was there anything else that was important that you were
Yeah,
trying
I
to
guess
answer?
I guess what I was at the point I was trying to get to about glycolysis is I think that because these are enzymes that in essence you turn them on and off with phosphorylation, that there's sort of biological sort of design principle hold on to them as long as you can, and if you want to turn them on or off, use phosphorylation to do that. And so I think that sort of contrasts with, like, let's say like signaling proteins, where oftentimes you do want to
Okay. Ooh.
Yeah.
abundant and where you just kind of want to hold on to them forever. That's where maybe there's less of an evolutionary driving force to make them refold and more of a driving force to keep them kinetically stable. At least that's my theory for it.
Okay, with that, maybe Caitlin, I'll turn it to you for any comments you would like to make, sort of to finish up our
To wrap
discussion.
everything up?
No, I'll wrap up at the end. But
Oh,
if
okay.
you wanted to add here, you
No,
have
I don't have anything else to say, I guess.
I think that all right, then I will wrap up, because there are more questions here. We may still get a chance to type a couple more. So I'd like to thank everybody for their stimulating questions. And this webinar will be posted also on YouTube, so it's a resource to keep thinking about these really important problems. I think this was really a great webinar. I'm really excited about the prospects here and all the new work that is coming out, because I think we have a lot more work to do. We've come a long way, but there's still now these new complexities of what in context means for protein folding and misfolding and assembly. And so, yeah, I'm really excited at the future prospects. Thank you all again for doing this.
Thank you.
Thank you.
I haven't answered everyone's questions, so if you want to email me, I could follow up.
Oh, Stephen, they're in the chat.
Yeah, but it'll disappear as soon as we close the session, right? Yeah, so if it's burning, you can send it to me in an email and I will get back to you.
Yeah, unfortunately, the chat questions don't end up on the YouTube thing, but further discussion is great. Thanks, Stephen. All right. Onward. Take care, everyone.
Okay,
Bye.
everyone.